So in this video, we're going to be discussing the TCA cycle. So we talked before about the process of glycolysis and how glucose is converted into pyruvate. We later discussed how pyruvate is converted into acetyl-CoA by the enzyme pyruvate dehydrogenase complex, and this is going to happen inside of the mitochondria. Acetyl-CoA can now enter the TCA cycle to produce more ATP. So in this video, we're going to be talking about the specific reactions that happen in the TCA cycle. Now, from the process of pyruvate all the way to the end of the TCA cycle, this all happens inside of the mitochondria. Okay, so now we're just going to discuss the TCA cycle in a bit of overview just to give you the general layout of this cycle. So we said that we get acetyl-CoA from pyruvate, and pyruvate is going to be the end product of glycolysis. Now, when acetyl-CoA or when pyruvate enters the mitochondria and is converted into acetyl-CoA, acetyl-CoA is going to react with oxaloacetate to form citrate. And this is going to be the first reaction in the TCA cycle. Citrate is going to enter a series of enzymatic reactions, and at the end it's going to be converted back into oxaloacetate. In the process, it's going to produce carbon dioxide, it's going to produce NADH, FADH, and GDP. Now, the NADH and the FADH2 are going to be transported to the electron transport chain, where they further produce more ATP. So this is kind of the general outline of the TCA cycle. Now let's talk about the reactions in more detail. So the first reaction, like we said, acetyl-CoA is going to react with oxaloacetate and form citrate. This is catalyzed by the enzyme citrate synthase. Now this is an irreversible reaction, meaning that it's going to be regulated. And the thing that's going to regulate citrate synthase is going to be ATP. When there's high levels of ATP inside the cell, this is going to inhibit citrate synthase. High levels of ATP indicate a high energy state, so there's going to be no need for the TCA cycle to go on. The second reaction is going to be the conversion of citrate into isocitrate. Now, as you can see, citrate has 6 carbons and isocitrate has 6 carbons. So they're just the carbons are just going to be structured differently. The enzyme that catalyzes this reaction is going to be aconitase, and this is a reversible reaction. The third reaction is going to be the conversion of isocitrate into alpha-ketoglutarate, and this is going to be catalyzed by the enzyme isocitrate dehydrogenase. Now, as you can see, isocitrate has 6 carbons, while alpha-ketoglutarate has 5 carbons. So the carbon that's going to be lost from isocitrate is going to be in the form of CO2. Another important thing to know is that dehydrogenase is going to be an enzymatic reaction in which a molecule gets oxidized. And when a molecule gets oxidized, that means it's going to lose an electron. Now, what's going to receive that electron? That's going to be NAD+. So NAD+ is going to receive that electron that was lost, and it's going to be converted into NADH. So, just as a general rule, any enzymatic reaction that contains dehydrogenase, there's going to be an NADH produced. Now, this is an irreversible reaction. That means that it's going to be highly regulated. The thing that's going to inhibit isocitrate dehydrogenase are going to be ATP and NADH. Now, ATP, of course, indicates a high energy state, so that's going to tend to inhibit the TCA cycle. So NADH is produced in the TCA cycle. So when we have a lot of NADH, that means that we don't need the TCA cycle to continue because we don't need to produce more NADH. Molecules that stimulate isocitrate dehydrogenase is going to be ADP. So when you have high levels of ADP, this indicates a low energy state inside of the cell. And of course, so this low ADP is going to stimulate isocitrate dehydrogenase and further stimulate the TCA cycle. The next reaction is going to be the conversion of alpha-ketoglutarate into succinyl-CoA. Now, alpha-ketoglutarate, like we said, has 5 carbons. Succinyl-CoA has 4 carbons, and it also has a CoA group attached to it. Now, this is going to be catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. And like we said before, anytime you see dehydrogenase reaction, there's going to be an NADH produced. And of course, alpha-ketoglutarate lost a carbon, so there's going to be a CO2 produced. Now, this is an irreversible reaction. And like we said before, any irreversible reaction is going to be regulated. So ATP and NADH are going, if they increase in the cell, they're going to inhibit alpha-ketoglutarate dehydrogenase. And succinyl-CoA, when its levels rise, is also going to have negative feedback inhibition on alpha-ketoglutarate dehydrogenase. Okay, so the next reaction in the TCA cycle is going to be the conversion of succinyl-CoA into succinate. Now what happens here is that succinyl-CoA just loses the CoA and we get succinate. There's going to be a release of a CoA group, and that's going to lead to the release of a little bit of energy in the form of GTP. This whole reaction is reversible, and it's catalyzed by succinyl-CoA synthase. The next reaction is going to be the conversion of succinate into fumarate. Now, this is going to be catalyzed by the enzyme succinate dehydrogenase. And remember that when we said when there's any dehydrogenase reaction, that means that there's going to be loss of electrons, and it's going to produce NAD+. NADH. But in this case, it's going to be FADH. The next reaction is going to be the conversion of fumarate to malate, and this is going to be catalyzed by the enzyme fumarase, and this is a reversible reaction. The last step is going to be the conversion of malate back to oxaloacetate. So like we said before, we start with oxaloacetate and we end with oxaloacetate. This is why it's called the TCA cycle. Now this is going to be catalyzed by malate dehydrogenase, and one NADH molecule is going to be produced.